Hi everyone, here we are today for Lecture 5, which covers Chapter 5, ABO and H blood group systems and secretor status. So to begin with our objectives, by the time we are finished with this unit, you should be able to define the ABO blood group system, blood group antigens and their inheritance, explain Landsteiner's rule, list the cells, body fluids, and secretions where ABO antigens can be located. Describe the relationships among the ABO, H, and SE genes. Moving on to our second page of objectives, you will be able to compare type 1 and type 2 oligosaccharide structures and locations for each. Describe the formation of the big H antigen from the gene product and its relationship to ABO expression. Discuss the selection of whole blood, red blood cells, and plasma products for transfusions. And list the glycosyl transferases and the immunodominant sugars for the A, B, O, and H alleles. And our last objective slide, compare and contrast the A1 and A2 phenotypes with regard to antigen structure and serologic testing. Describe how to differentiate among A3, AX, and AL subgroups by serologic testing. Determine the possible ABO genotypes based on ABO phenotype. And describe the ABO blood group system antibodies with regard to immunoglobulin class, clinical significance, and in vitro serologic reactions. Oh, I was wrong, sorry. One more page of objectives. Define the terms universal donor and universal recipient as they apply to both red cells and plasma products. List technical errors that may result in an ABO discrepancy. Define the acquired B antigen, the BA phenotype, and discuss ABO discrepancies that result from these phenotypes and methods used to resolve them. And one more. List reasons for missing or weakly expressed ABO antigens and the test methods used to resolve discrepancies such as these. Describe ABO discrepancies caused by extra reactions in serum testing, which is reverse typing, and how they can be resolved. Discuss the Bombay phenotype, its genetic pathway, serologic reactions, and transfusion implications, and define the terms secretor and non-secretor, and that should really be it for objectives. Okay, so you all had an introduction to the ABO system in Module 1 through our labs, and so a lot of this stuff is going to be information you've previously heard. So with the history, you all know Carl Landsteiner was a scientist who discovered the ABO blood group system in 1900. And he came up with a rule called Landsteiner's rule or Landsteiner's law. And his rule states that healthy individuals possess naturally occurring ABO antibodies to ABO blood group antigens that are absent from their red cells. And so we've talked about this previously. Here's what's important. They are naturally occurring. So an individual does not need to have a transfusion or a pregnancy to develop these antibodies. They develop on their own. And that's really critical to remember. This slide shows the relationship between ABO antigens and antibodies. And we reviewed this a lot in labs 1 and 2. So this is a diagram from our textbook that describes this relationship once again. And so, we know that the testing that we do in the lab, we determine a phenotype. And so when someone is AB, it means they have both the A and the B antigen on their red cells. If you think about Landsteiner's rule, once again, they would only have an antibody to an antigen lacking, not present on their red cells. So in this case, since they have both A and B, there are no ABO antibodies. And so this is why in the past or I may have mentioned to you in lab, that type AB is actually the universal plasma donor, because giving someone type AB plasma, there aren't any ABO antibodies that can cause harm. When we move on to people who are type A, we see they just have the A antigen. So think about, what don't they have? They don't have B. So they're going to have an anti-B. When we move on to type B, we see they have the B antigen. What don't they have? A. So they're going to have anti-A antibodies. And then someone who is type O does not have any antigens on their red cells, so they are called the universal red cell donor. What this means, Landsteiner's rule, they're going to have both anti-A and anti-B in their plasma. So moving on to this next slide, general characteristics. ABO antigens are an intrinsic part of the red cell membrane, but what you may not know from our previous discussions is that they can also be found in body fluids soluble. And so today we're going to be looking at how that occurs. ABO antigens are detected in the developing embryo as early as 5 to 6 weeks gestation, so they form pretty early on. Newborns have fewer numbers and only partially developed antigens and won't have full ABO antigen expression until between 2 to 4 years of age. So while we can detect antigens on newborns, and it's an important part of newborn testing to every baby that's born, full expression does not occur until they're a little bit older. Here we see table 5.2 from our textbook, and this is data that basically breaks down different ABO phenotypes, on 3 races. So we see whites, blacks, and Asians, and I apologize that there's not a Latin race here. This is just what was available from our textbook. But what's interesting, if you look at the 3 races that are listed here, you can see that type O among all of the races is the most prevalent, okay? And so O is actually the most common blood group, no matter what race you happen to be. When we move on to type A, we could see that A is second in all 3. But we can see that there's definitely a higher incidence of A in whites than, let's say, blacks and Asians. When we move on to type B, I find this really very fascinating. B is the 3rd most common blood type, no matter how you cut it. However, when you look at people of Asian descent, you can see that the B is much more prevalent in them, almost as prevalent as type A is, okay, people of Asian backgrounds. And then lastly, type AB, no matter where we're looking, it is the rarest blood type among all 3 here. Okay, so sorry I don't have additional information for other ethnic backgrounds and races here, but I'll be presenting some more stuff to you guys as we move on that I found from other sources. So let's talk about ABO and big H antigen. So there are genes at 3 separate loci that influence the occurrence and location of ABO antigens. And so ABO is absolutely linked to something called the big H, and it's also linked to something capital S, little e, which I'll call SE, okay? So the presence or absence of A, B, and H antigens on the red cell membrane is controlled by the H gene. And notice it's not just H. This H gene controls A and B antigen expression as well. The presence or absence of ABH antigens in secretions, such as saliva and body fluids, is influenced by a separate gene called the big SE, and this stands for secretor, okay? Which is nice and logical because, it rules what antigens we find in secretions. So these are soluble, not attached to a red cell membrane. Now here's where it can get a little bit confusing, so bear with me. We're first going to start with big H gene, okay? So big H does code for expression of an antigen, And they designate little h as its counterpart. However, little h is what a term we've looked at before called an amorph, meaning it has no detectable gene product. Okay, so why are we looking at big H? You need to inherit at least one big H from your mother or your father in order to have A, B, and or H antigens expressed on your red cell membranes. Okay, so it is linked to whether A and B can be there as well. There are some rare individuals who we will look at towards the end of this lecture called the Bombay phenotype, and they're named that because this was first discovered in India, in Bombay, which I believe now is called Mumbai. But these individuals lack the H antigen. Okay, so what does that mean as far as inheritance? They are homozygous for little h. Okay, so little h, they have no big H to confer the antigen, and therefore they are not able to have A or B antigens either. Okay, so what you want to take away from this is you absolutely need to have inherited at least one big H in order to express A and B antigens. Without that big H, even if you inherited A and or B alleles, those antigens won't be expressed on the red cells. So when we move on to the secretor gene, okay, big SE is kind of like the big H, okay, except remember this is related to secretions like saliva, and there's a little SE, and so you can see how I have a line that I put over little SE, and the reason why I did this is because small case s and big case s look very similar, so when you see a line over it, you know it's the smaller case. So just like little h is an amorph with no detectable gene product, little SE is an amorph, okay, to big SE, so there's no detectable gene product from little SE. So you need to inherit at least one big case SE allele to express A, B, and H antigens in secretions. Okay, so this is strictly related to secretions. Okay, so when we have an individual who inherited small case SE, small case SE, as we see here, they will not have A, B, or H antigens in their secretions. Okay, regardless of if they have big H. Okay, so just remember secretions are ruled by this big SE gene. Our ABO genes, remember A, B, and O alleles are located on chromosome number 9, and they exhibit codominance. Okay, so it's possible, that's how it's possible for us to have type AB or type A or type B or what have you. Okay, so hopefully that makes sense. So let's move on and look at the structures of these oligosaccharide chains. So the oligosaccharide chain is a basic precursor structure for several red cell antigens, including A, B, and H. And it's attached to a protein or a lipid carrier molecule. And so this is a diagram from our textbook. And what's important to note here, some of you have had organic chemistry maybe in the summertime, or previously I know you've had it. Not one of my favorite topics. However, we can see here a type 1 oligosaccharide chain, and we see different carbohydrates linked together. And this is what, this type 1 chain is what we find in body fluids and secretions. Okay, so it's somewhat different than the type 2 oligosaccharide chain, which is what we see mostly on red blood cells. Okay, so a little bit different structure. Okay, so when someone refers to a type 1 chain, that's strictly related to fluids and secretions. Type 2 is where this type of chain is used for our red cells. Can be present also, though, in body fluids and secretions, but you see red cells are not associated with a type 1 chain. So let's talk a little bit about how these antigens are constructed. And what's very, very important to remember is that inheriting these alleles doesn't automatically mean you have the antigen. There's much more involved. Okay, so it's really chemistry. We're going to look at big H first. So the big H gene, remember, you have to have at least one. It actually codes for an enzyme, a glucosyltransferase. And so when we look at this type of enzyme, a transferase, this is an enzyme that transfers a certain sugar, which we'll call immunodominant sugar, to our basic oligosaccharide chain, our type 2 chain, to form the H antigen. Okay? So this glucosyltransferase that the big H gene codes for actually transfers a sugar called L-fucose. Okay? And so remember, when you inherit the gene, okay, it's coding for an enzyme, okay? And this L-fucosyltransferase is going to take L-fucose sugar and add it to this type 2 oligosaccharide chain. So we saw this on the last slide, right? This is our type 2 chain. And so when you inherit at least this one big H, this glucosyltransferase enzyme is going to transfer L-fucose here, okay? And so this is the building block. We need to have this L-fucose in place on the type 2 oligosaccharide chain for A and B antigens to come next, okay? So, and that's what the second bullet says. So the H antigen is the foundation for A and B antigens. If we don't have this L-fucose in place, even if you inherit A and or B alleles, nothing can be added, okay, because they have to be added to L-fucose, all right? And so here in this picture, and I'll talk about it on the next slide, we see, for instance, the A antigen. So when you inherit at least one A allele, okay, here's what it does. It codes for another enzyme, okay? The gene product, it's not the same enzyme as big H. It's a different transferase enzyme called N-acetylgalactosamynyl transferase. It's a mouthful. But this transferase enzyme is going to transfer this sugar, N-acetylgalactosamine, and it's going to place it onto the L-fucose building block that we started with the big H, okay? So you look here, here's our building block H, and now when you have the A gene, okay, it's going to add N-acetylgalactosamine, that's the abbreviation for that, added, okay, to the structure that we have from the H antigen. Okay? When you inherit a B allele, a different enzyme is coded for. It's a D-galactosyltransferase. Okay, so what sugar does it transfer? D-galactose. Okay, so instead of transferring N-acetylgalactosamine, we're going to have D-galactose added to the L-fucose H building block. Okay? If we were meeting face to face, I'd be asking you guys this question right now, and I'm sorry you can't answer it for me, but here we go. If you don't have L-fucose, if you don't inherit at least one big H, can you have A or B? The answer is no, okay, because we need to have that building block of the L-fucose there for the sugars that are associated with each of these different alleles to add to the building block to make the antigen. Okay, so this next slide basically sums up what I described on the last slide with the picture. So just remember, A gene codes for a different transferase enzyme, the N-acetylgalactosamine, which is going to add N-acetylgalactose amine to basically the H antigen. I'm sorry, it's an N-acetylgalactosamynyl transferase. The gene product is always an enzyme. The B gene codes for D-galactosyltransferase. It's going to transfer D-galactose to the L-fucose building block. When someone has type AB, okay, they have both enzymes, okay, so they're going to have both of these sugars added to L-fucose. All right, so we're going to move on and talk next about antigen concentration. So with regard to big H antigen, remember we have to have it in order to express A and or B or both. there are certain blood types that have more H antigen than others. And, hopefully this first one is going to make sense. Group O individuals actually have the most H antigen sites. Okay? So Group O has the most. And let's think about this for a minute. When someone inherits 2 O alleles and they're type O, remember O is an amorph. It really doesn't code for an enzyme to transfer any sugar to the L-fucose. So it would make sense that they have a lot of H antigen sites because it hasn't been modified into an A or a B antigen, okay? Because O is an amorph. It doesn't code for a detectable gene product. The next one with the most binding sites of H, or H antigens, I should say, is A2, followed by B. Then we have A2B, A1, and A1B with the least. Okay, so we're going to move away from big H. Hopefully that makes sense. And we're going to talk about subgroups of type A. So when an individual is group A or type A, the two main groups that we see are A1 and A2. And so somebody could be A1 red cells or somebody could be A2 red cells. When we're doing our forward typing, whether you're A1 or A2, both of those antigens are going to react strongly with our antibody against A reagent, or anti-A, and give 3+ to 4+ reactions. Okay? And so basically, if you're curious, 80% of the population of people who are A or AB are type A1, and 20% are A2 or A2B. Okay? And the antigens themselves are different. And so this is, once again, another diagram from our textbook. But here, when you look at A1 phenotype, we can see that the A1 antigen is branched, kind of looks like a little pitchfork or something. And A2, and this is really simplified, the A2 is more linear, okay? So they're slightly different antigens. They have a different structure to them, okay? So how can we tell when we have a type A individual if they're A1 or A2? You may recall maybe from lecture 2 and 3 when we were talking about blood banking reagents. There is a lectin which comes from a plant, the plant Dolichos biflorus, called anti-A1. And this lectin acts like an antibody against the A1 antigen. Okay, so if we wanted to see if someone was type A1 or not, we would take their red cells, their 2 to 5% red blood cells, and add a drop of A1 lectin to their red cells and see if their red cells agglutinate. Now, if they agglutinate, it means that they are type A1, okay, because this works the same way as an antibody against A1. So agglutination means you're A1. If the red cells don't agglutinate, most likely it's type A2, okay, because that is 20% of the population. Okay, and so, we do have some people in our region who are type A2. It really does not impact transfusion, okay? And remember, they're both going to react strongly with anti-A in our forward typing. So this is table 5.4, looking at phenotypes and possible genotypes. Let me go back 1 minute. I don't know if I mentioned this, but A1 is actually dominant over A2. So here we see if someone is A1, they could be A1, 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 A2, because A1 kind of dominates over A2 or A1O. To be group A2, you have to be either A2, A2, or A2O, okay? And then, A1B, of course, we only get one allele from each parent, and A2B. Okay, so in this case, this is one instance in the ABO system where these two are not co-dominant. Okay, so A1 is actually dominant over A2, so the A1 is going to be expressed. Okay, so beyond A1 and A2, there are some very rare subgroups of type A, and we're going to mention them briefly here. There's one, and they usually write them as a subscript in the bottom here. So there's A intermediate, A3, AX, AM, AN, AL, A Bantu. And they don't come up very often, but we want to suspect that a patient may have a rare subgroup of type A if a couple of things are happening. Okay, firstly, when we're doing our forward typing, we see weak agglutination or no agglutination with anti-A and our combo anti-AB reagents. Okay, so either nothing or weak. Okay. When we look at reverse typing, the patient has an antibody against A1 cells. Okay, and so that means, think about reverse typing, that means that their plasma is agglutinating the reagent A1 cells. We have an anti-H lectin from a plant, Ulex europaeus. Anti-H lectin strongly agglutinates the patient red cells. We want to suspect it's a rare subgroup of A. Okay. Usually these come up because there's a discrepancy between forward and reverse typing. Okay. And so we're going to be looking more carefully at these reactions a little bit later. But for now, just remember that they're rare. Okay, so they're not common. There are some subgroups of type B, but they're much less common even than the subgroups of A. And typically, they would not react strongly in forward typing with anti-B, like we would expect someone who is a type B to react, 3+ or 4+. They react more weakly. So these subgroups are usually picked up by a blood donor center who is testing lots of different donors, not usually by hospital labs. So let's talk about why it may be important to identify when someone has one of these rare subgroups. Why is it important? If a weak subgroup is missed, let's say somebody is one of those rare A subgroups like AX, AINT, or what have you. If we miss it in a donor, okay, remember donor red cells, our red cells are going to be transfused into a recipient, okay? If we miss that subgroup in forward grouping, there's no reaction, that donor may type as O, okay? And what do we know about type O? Type O is the universal donor. Okay, so we could be giving erroneously these A subgroup cells, which we think are O, to a recipient, and there could be a transfusion reaction. Okay, so here's my example. I'll go over this again. So let's say we have AINT or AX or A Bantu or one of these, and it's missed in the donor, meaning there's weak or no reaction on forward typing, so they're going to call them an O. O is universal red cell donor. We might give group O to a group O recipient because remember, O can only get O. So they would be receiving a unit that really is not group O, has this rare group A antigen on it, and they could have a transfusion reaction because think about it. Landsteiner's rule, someone who is type O has naturally occurring anti-A and anti-B. So when we give that rare subgroup of A to this recipient, their anti-A is going to link on to that antigen and there will be a transfusion reaction. So that's the significance of finding out. Okay, so we're going to move on and talk about ABO system antibodies, which we did in lab 2 a little bit already. So as I mentioned at the beginning of this unit, we have to remember that these are not red blood cell stimulated, meaning not due to being exposed to another's red cells. Okay, they're naturally occurring. And this actually happens, due to exposure to substances in the environment that are similar to A and B antigens, bacteria. Okay, so we just basically, have these naturally occurring antibodies. ABO antibodies are not usually detected until 3 to 6 months of age. And titers or concentrations reach maximum levels by 5 to 10 years of age. Here's something important. As individuals get older, into their 70s and 80s, the amount of antibody actually can decline. And in general, okay, that can happen with people as they get older. All antibody levels can decline. Okay, so hopefully this is going to look familiar. So here we have our blood groups and the antibodies. Okay, so someone who is blood group A has anti-B. It's mostly IgM with some IgG. Someone who is blood group B has anti-A, mostly IgM and some IgG. And then someone who is type O, they're interesting. They have anti-A and anti-B, and then they have that combo antibody AB, and this is mostly IgG. Okay, and remember, you don't see type AB up here because would type AB have any antibodies? No. Okay. So anti-A and anti-B antibodies in group A or B individuals are mostly IgM. IgM antibodies are clinically significant because they are very, think back to module 1, they are very readily able to bind complement. They react optimally at immediate spin testing in cross-matching. And cross-matching is a test where we take donor red blood cells and combine them with the recipient's plasma to see basically if they're compatible. And we try to do that prior to transfusing the recipient with those red cells. And we'll be talking more about cross-matching later. This anti-AB antibody is found really only in group O individuals, and it will cross-react with both A and B antigens. So let's talk next about anti-A1 antibody. So a few slides ago, we talked about how when you're type A, you could be type A1 or you could be type A2. So individuals who are not type A1, other subgroups of A, A2, and potentially A-int and all of those other ones, they have a different antigen structure. And so it's quite possible that they can develop an antibody specifically against the A1 antigen because A2, even when we looked at the picture, the antigen structure is different. These anti-A1 antibodies are not clinically significant, meaning that they're not related to any kind of transfusion reaction. However, they can cause incompatible cross matches with immediate spin testing, and they absolutely can cause discrepancies in reverse typing. So think about reverse typing. That's when we take the patient's plasma and we test it with reagent red cells, right? We test with A1 red cells and B cells. So if they happen to have this anti-A1, it's definitely going to link onto the antigen of our A1 red cells. Okay, and so it's going to look like a discrepancy in typing. Okay, and we're going to go over these discrepancies in more detail at the end of this unit. All right, so next we're going to review ABO phenotyping, and this should be really easy because you've heard me talk about it ad nauseum by now in labs 1 and 2. So when we're doing our ABO phenotyping, remember forward grouping is when we are determining which antigens, if any, are on the patient's red cells, and we use the known antisera. This determines the ABO phenotype. As a double check, we do reverse typing or reverse grouping, where we take the patient's serum or plasma and we test it with reagent red blood cells, A1 cells and B cells, to see which ABO antibodies, if any, the person has. And this is really done as a double check of our forward typing. Okay, we want to make sure that if the typing going forward looks like O, that the reverse typing looks like O2, meaning they have anti-A and anti-B that reacted with the reagent A1 and B cells. Both forward and reverse testing are required on donor and recipient blood samples, and this is stipulated by standards for blood banks and transfusion services. Have to do both. So here we see table 5.6, ABO phenotype reactions, and hopefully by now some of this is making sense to you. So cut it here. When we have group A, they have A antigen. It's going to be positive with anti-A. What antibody does group A have? Anti-B. And anti-B should be positive with the B cells. Okay? Excuse me. When we have group B, they have B antigen, which should be detected with our anti-B. They should have anti-A, which is going to be detected by our A1 cells. Group O, we have no antigens, which means we should get reactivity with both A1 and B cells because they have anti-A and anti-B. Group AB has both antigens, which means no antibodies. Very important. Commit that to memory. So let's talk about which kind of units we select for transfusion. So we'll start with red blood cell transfusion. So packed red blood cells are the most common product that is transfused. ABO identical unit is preferred. What does that mean? The same type. So if you're type B, we want to give you type B. That's always best. Okay? Followed by ABO compatible unit. Okay, so try to go with the same ABO blood type first. If we don't have a unit in our refrigerator that is the same type, then we can go with one that's compatible. Incompatible red cells can cause an acute hemolytic transfusion reaction, and we will study these transfusion reactions in an upcoming unit. They are most often fatal within a few minutes. Okay, so no mistakes allowed. When we are transfusing whole blood to an individual, they can only get identical type, the same type. Here's why. Think about it. If you're getting whole blood, you're not only getting red cells, you're getting plasma, you're getting everything together. So you're only going to go with the same exact ABO type. So if I'm type A, I need to get type A whole blood. No other thing is going to work. Okay? As I mentioned to you all earlier, group O is the universal donor for red cells. Okay? When we're talking about recipient of red cells, group AB could receive the red cells from anybody. They could get A red cells because they have the A antigen. They could get B red cells because they have the B antigen. They could get type identical, which is AB, or they could get O, the universal donor. So let's switch our thinking and move on to plasma transfusion. Okay? So similarly, ABO identical unit is always preferred. Okay? Give the same type. If we can't, we want to make sure it's compatible, and we want to consider the reverse, okay, of red cell transfusions. Okay? So what does that mean? Type O is not the universal plasma donor. It's actually type AB. So think about it. We want to give the type that lacks any antibodies. Okay, so for plasma, type AB is the universal donor. And in this case, type O is the universal plasma recipient because type O has both anti-A and anti-B. So receiving those antibodies in a plasma transfusion won't cause harm. Donor unit antibodies must be compatible with the recipient's red cells. Okay. So this is basically the summary, table 5.7. And so we see all of our phenotypes. And to reiterate, cannot stress it enough. If someone is getting a whole blood transfusion, it has to be the same blood type. No other choice. That's it. When we look at red cells, remember, O is the universal red cell donor. You see, everybody could get O. You can always get your own type. Okay? So we can see type. Remember, AB is the universal red cell recipient, and so they could get AB, AB, or O. And if you're group O, you can only get O. When we're thinking about plasma, we want to be worried about the antibodies that we're giving to somebody. And so remember, AB is the universal plasma donor. So you see that is the one that is listed here for everybody. So you can always get your own type, and you can get AB, okay? If you happen to be type AB, you can only get AB. And if you happen to be type O, they have both anti-A and anti-B antibodies, so they could really get, they are the universal plasma recipient. So this is really a summary of the last slide. So let's move on and talk a bit about ABO discrepancies. An ABO discrepancy is when we do our forward typing and we think that the patient, is one type based on the antigens we're picking up, but then the reverse typing, the antibodies that they have don't fit, so it's a mismatch. Discrepancy can be present. Okay, what are we going to look for? How do we know which reaction is the one to give us a clue about what's going on? We know that the ABO reactions are usually pretty strong, 3+, 4+. If we see one of our tubes has a weaker than expected reaction with agglutination, that might be the one that's discrepant. Sometimes a reaction we're expecting to be positive is negative, so it's missing. Or sometimes there are extra reactions that show up in our testing, and we'll be looking at all of these in a few minutes. So when we have a discrepancy, technical considerations. First of all, we want to first start with, was there a mistake made with sample identification on the tubes? Did I accidentally label something wrong? Did I put, the wrong plasma or person's red cells in the tubes? Maybe the results aren't properly recorded. Maybe I didn't interpret my results accurately and properly record them. So, when we're looking at in labs some of the YouTube videos about, reading tube, you can't overshake the tube, okay? So ABO reactions are usually pretty strong, but when we move into other red blood cell antigen systems, those reactions can be weaker. So we have to be very careful not to overshake our tubes. Was quality control done, and was it satisfactory on our reagents? Is there contamination or hemolysis of our reagent red cells? Did we centrifuge for the appropriate time, and was our centrifuge calibrated? And then did we follow the procedures? Add the appropriate number of drops, make the proper red cell suspension, our 2 to 5%, completely suspend our buttons. So all of this, has to be taken into consideration. Okay, if we realize that, it's not due to any of these technical discrepancies, then we have to move on and look at sample-related discrepancies that could be causing the issue. And so this is really an overview, table 5.9 from our textbook, of some things that we're going to be looking at individually. So first of all, sometimes in our forward typing, extra antigens show up. Okay, and so we see a bunch of things listed here. In our reverse typing, sometimes extra antibodies show up. Okay, sometimes we have missing or weak reactions in both antigens and antibodies, and mixed field. Okay, so mixed field, I may have mentioned to you guys previously. I'm just going to take a minute to talk about that. Mixed field reactions, when you're doing your tube testing and you resuspend the button, it looks like you have some cells agglutinating in there, but some are, looking smooth. Okay, so it almost looks like you have a positive and a negative in the same tube. So we call that mixed field. So we're going to jump right in and talk about some of the more common ones. And so this slide has to do with extra antigens that we're picking up in our testing. And so we have to think, where are patient antigens detected in forward typing? Okay, so these would be extra reactions in our forward typing. So the first one This is a person who is really group A with acquired B, okay? So when we're doing the testing, it actually looks, you know, they're reacting with anti-A strongly, but then there's another reaction that's showing up that makes it look like they also have a B antigen, and that's usually a weaker reaction, okay? So why does this happen? The group A immunodominant sugar is altered by a bacterial deacetylating enzyme. So it basically breaks down, you know, the A antigen, so it looks like a B. Okay, so when does this happen? Some group A individuals who have a lower GI disease, obstruction, or gram-negative sepsis. So knowing patient history is going to be important too. And remember, it looks like they've acquired a B antigen, so they're going to show reactivity with anti-B in forward typing. And so they actually look like they're AB, okay? And, you know, we will look at all of these, the test results of what we would expect a little bit later. So for now, I'm just going to describe them, and we will look at what actually happens with them in a minute. Let's see. Okay, BA phenotype. It's very similar to this one, except in this case, we have an individual who is type B, and it looks like they've acquired an A antigen. So this is similar to acquired B. Okay, as I said, however, the patient is group B, and they're showing reactivity with anti-A. So once again, they're going to look like they're a type AB. Okay. Oops, sorry, wrong way. Okay. Extra antigens also in forward testing. Polyagglutination. You guys know that prefix poly means many, many red cells are agglutinating. And so here, there can be a hidden antigen on the red cells that's exposed and reacts with most human sera. So it's non-specific agglutination that's happening usually in all of the tubes. So this non-specific aggregation could be due to a couple of things, and you've heard about this first one before. Our favorite, one of our favorites, rouleau. Remember those stacks of coins? So when someone has an abnormal amount of proteins present in the blood, remember the proteins can coat the red cells and form rouleau. And you know, if there's an extensive amount of rouleau, it could look like weak agglutination reactions in our tube testing. The next thing, Wharton's jelly, is something that specifically is found in cord blood. And cord blood is from the umbilical cord of a newborn. And so when babies are first born, they take blood from the umbilical cord and they send it to the blood bank, and we test it. We only do forward typing on it, and to find out the baby's ABO and RH type. So this Wharton's jelly is really gelatinous, and it could be very gooey and viscous, and so it can actually cause, you know, it can look like red cells are aggregating, but they're really not. It's due to the Wharton's jelly. Okay, so I think we're going to do a couple more, and then I'm going to jump ahead and we'll look at the reactions. Okay. So ABO subgroups, okay, we talked about A subgroups a little bit earlier, and you know, we said how some of the very rare subgroups can demonstrate weak or no reactivity with anti-A, or if it's a subgroup of B with anti-B. Sometimes chronic conditions that a person has can affect this as well. So patients with leukemia or Hodgkin's disease may show weakened A or B antigen expression, so we may be getting very weak reactions in the forward typing. So it's very important to check the patient's diagnosis and transfusion history, and if need be, we could repeat with our combo reagent anti-AB to enhance subgroups and possibly incubate at room temperature before centrifuging and reading. So if we have these weak reactions, we could add our anti-sera to the tubes, add the red cells to the tubes, but instead of doing immediate spin, centrifuging right away and reading, we could let them sit at room temperature for 10 to 15 minutes and give antigen and antibody time to come together, and then look at them. And sometimes that will make the reactions a little bit stronger. Okay, so I am going to stop my share for 1 minute. Oops, and I'm going to try to jump ahead. I want to see if I could show you, since we talked about some of the forward typing things, I want to see if I could show you. Okay. And this is what I want to show. So let me just make this bigger. Okay. So this is a couple slides ahead. Oh, no, it's the wrong one. Oh. Sorry, guys. Bear with me. Okay, this is the one. So we talked about how some of these reactions can show up as extra reactions in our forward typing. So we're going to look at some of the examples that we talked about. So the first one here, this discrepancy, acquired B. Okay, let's look at what we have. So we have our forward typing results here and our reverse typing results here. So remember, forward typing detects antigens present on the red cells. Reverse typing detects naturally occurring ABO antibodies. So when someone has acquired B, okay, let's look at it. Here we see this very strong 4+ reaction with anti-A, okay? However, we see a reaction here, 1+ with anti-B, okay? So right now, if we were just looking at the forward typing alone, we would have to say the type here looks like AB, okay? So let's look at our reverse typing. We look at reverse typing, we see no reaction with A1 cells, which means they do not have an antibody against A1. And we see a strong reaction with B cells, so they have an anti-B, okay? So forward typing tells us they look like AB, but reverse typing tells us they look like A, okay, because they have this anti-B that's reacting. So that's a discrepancy. So which reaction is the one that we're going to be suspicious of? Remember, one of the things you want to look for is if there is a reaction that is much weaker than the others. And so when we look to this anti-B, we can see 1+, well, that's really weak for ABO reactions. And so that's the one that we want to think might be discrepant. So think about it. If we eliminated this 1+ reaction and this turned into a negative, would the forward and the reverse typing then agree? And the answer is yes, because if this were eliminated to a negative, this patient would type as A on forward and type as A on reverse. Okay, so this is the reaction that looks discrepant. So how are we going to resolve this? We are going to test this patient with autologous red cells. Okay, so what does that mean? We're going to take this patient's serum and we're going to test them with their own red cells. Okay, we know they have anti-B here. Okay, if they really had a B antigen, they would be agglutinating their own cells, and they're not going to be, okay, because basically this is a false reaction. So this is basically patient control. So we're going to take the patient's serum with their own red cells. If they really don't have the B antigen, we're going to see a negative result, and it's going to prove that this is acquired B. Okay. We'd also want to maybe look at the patient's history to see if they have lower GI disease or something that could be causing the acquired B. Let's look at the next one. Okay, the BA phenotype. So let's look at our forward typing. Okay, we see a 1+ reaction with anti-A, so it looks like they have A antigen. 4+ reaction with B looks like B antigen. So here, forward typing is once again looking like AB. However, when we look at reverse typing, we see they have a strong anti-A and no anti-B. So it looks like they're typing on reverse as B. So A, B, and B. Okay. So once again, we want to look to see, is there a reaction that's weaker than the others? And we're going to look to this one, the anti-A. So think, if this reaction were eliminated to a negative, would forward and reverse typing then agree? And the answer is yes, because if this were negative, they would forward type as B and reverse type as B with anti-A. Okay? So how can we resolve this? We can test with a monoclonal anti-A from another manufacturer. And so sometimes, you know, the reagent, if we test with a different anti-A, this reaction here is going to go away and turn into a negative, and then discrepancy resolved. Okay, so this is really a person who's type B who looks like they have an A antigen. So our next one, subgroup of A, let's look at this. We see no reaction with anti-A and no reaction with anti-B. So forward typing looks like a type O. However, when we look at reverse, we see strong reaction here, meaning they have an antibody against B, which means they're type A. Okay, so why? One of those rare subgroups of A, remember, they may not always react with our anti-A, and so we want to try to strengthen the possibility of that reaction. If something showed up here, would it make sense then? And the answer is yes. So here we have a missing reaction that we expect, and so what we want to do is extend the incubation of these tubes. Like, let them sit for 10 to 15 minutes at room temperature before we centrifuge them. And we also could repeat with anti-AB. And sometimes this reagent, the antibody is a little bit stronger. It's going to detect those subgroups of A. Okay. So I'm going to come back to this one because I didn't talk about that slide. So bear with me. We're going to go back to slide 28. Yes. And talk about mixed field, and then we'll return. Oh, wrong one. Sorry, guys. My mouse is breaking down. My mouse has seen better days, unfortunately. Okay, there we are. So we're going to talk about mixed field, and then we'll look at the last. We'll go back to the chart and look at how it would look as far as results. So I mentioned to you guys, when you have mixed field reactions going on, you're seeing red cells agglutinated in the tube along with unagglutinated red cells. And these are pretty big agglutinates. So it's not like a 1+ where there are tiny agglutinates with a turbid background. They're a little bit bigger. So what can cause mixed field reactions to occur? The first thing that you really want to think of is 2 different red cell populations are present in this person. So it could be an individual who was transfused with group O red cells. So remember, group O is our universal donor, but maybe this person's blood group is AB or even AB. So, you know, the O red cells that are present in there may not be agglutinating with anti-A, but the A person's red cells are, that sort of thing. Okay. Someone who's had a bone marrow transplant. When someone has a bone marrow transplant from a donor, not autologous, their ABO blood group can change, and sometimes it takes some time for that transition. If someone has had a stem cell transplant, there's an A3 phenotype that can cause this, or T and polyagglutinable red cells. Okay, these are red cells that just agglutinate haphazardly. I'm just going to jump ahead this way. Bear with me. Okay. So here's our chart that we looked at, these 3 already. And so let's look at what might happen in a case of a person who's type B who was transfused with group O red cells. So when we look at our forward typing, we see no reaction with anti-A, so no A antigen. However, the anti-B tube has 2+ MF mixed field. So what this means is, you know, some of the red cells are agglutinating here and some of them are in a smooth suspension, okay? When we look at reverse typing, okay, it still looks like they're a type B. It's just we have this mixed field here. We want to find out what's going on with that. We see strong reaction with anti-A. That makes sense for someone who's type B, right? And no reaction with the B cells. So they back type or reverse type as type B, and they front type as type B. But what is the mixed field about? We want to check their transfusion history. And so sometimes this can be tricky, especially if you're working at a trauma center where maybe somebody is transferred from a community hospital and they had the transfusion at the community hospital, and then they're transferred into your hospital and you don't have all of the information as to what happened to them previously. But let's look at what's happening in this tube. So this is someone who's type B. So their B cells, okay, their B antigen is reacting with the anti-B. However, the donor group O red cells that they were transfused with are also in their circulation. And the O are the cells that are not reacting with anti-B because O has no antigen. Okay, so that is basically what's going on there. Okay. So we're going to move on and talk about discrepancies that arise due to extra antibodies. So these are going to show up in our back typing or reverse typing. So we can have extra agglutination reactions in our reverse testing. So let's talk about some things. So first of all, let's say we have a subgroup of A that's not A1. Remember, the antigen structure is different, and so these subgroups of A can have a naturally occurring antibody against A1, and that could show up as, you know, obviously agglutination with A1 red cells. Sometimes people can have cold ALO antibodies, and so a cold antibody is one that reacts best at room temperature, and room temperature is where we do our ABO testing. And these are antibodies that are directed against certain red cell antigens that react at room temperature. We can also have cold autoantibodies, and these are antibodies directed against self-antigens that react best at room temperature. Okay, and then we can have rouleau. Okay, so we talked about rouleau before. Remember rouleau, the red cells are coated with proteins. They make the stacks of coins. This could show up as weak agglutination reaction. Okay, so let's say we suspect that our patient has rouleau. And so the rouleau, those extra proteins in the patient's plasma are basically, you know, going to be coating our reagent red cells in reverse typing. So what can we do to try to resolve rouleau? So, you know, after we have the patient's serum and the red cells, we're going to centrifuge for 1 minute. And you're going to say, 1 minute? We usually only centrifuge for 15 seconds. And you'd be right. We're going to centrifuge for a longer amount of. After this centrifugation, we're going to take a pipette and remove the serum or plasma from the tube and discard it. Okay? So, you know, we combine everything and we incubate it to allow antibody to find antigen. We centrifuge, and then we're going to remove the serum or plasma and get rid of it. That's where all those extra proteins are. We're going to replace that serum or plasma with an equal amount of physiologic saline and mix it up. It's kind of like washing cells, okay, right? Now we're going to centrifuge for 15 seconds, and we're gently going to resuspend the button and read. So if after, you know, removing the serum and adding the saline, now we see no agglutination, we know that, you know, that initial agglutination we saw was false. It was due to rouleau because the saline basically, you know, washed away proteins and removed, you know, that false reaction. If we still see agglutination, we can say now this is true agglutination because immune complexes did form and were present in the tube, and they remained even after we washed with the saline. Okay, so that's the idea behind it. Okay, so the next slide, we're going to get into missing or weak antibodies in reverse typing. So it's going to show us weak reactions or negative agglutination there. So reactions may be explained by investigating patient history, age, diagnosis, and immunoglobulin levels. So firstly, we have to look at age, and we need to remember that the very young and the very old have reduced amounts of antibodies, all antibodies, okay? And so age could have something to do with these weak or negative reactions. Pathologic states that lower immunoglobulin levels, someone who's immunocompromised, could also be at stake. So knowing a little bit about the patient history can also help. Okay. So here's our survival tip for discrepancies. We first want to rule out technical errors because most often we humans are human and it is a technical error. Okay. So strategy 1, repeat ABO typing on the same sample to exclude technical error during testing. Wash red cells several times with saline, suspend in plasma. Okay. We could test a new sample, get a new sample if we can't resolve it. Okay. If it remains, we always want to gather history about the patient. And then we want to think weak, missing, extra. Okay? So we looked at this slide already. So we're going to look at the next one. Okay, and this is going to outline some of the other discrepancies we just talked about with reverse typing. So our first one is a group A2 individual who has anti-A1. So what do we expect? Let's look at our forward typing. So the forward typing, they type as A. Okay, that's fine. We look at reverse typing, however, it looks like they have an anti-A1 and an anti-B, which means they're looking like type O on reverse typing. So that is absolutely a discrepancy. So what can we do? Remember, we want to look for extra reactions that might be weaker. 4+, 4+, there's the culprit. So this is a weaker reaction, and we want to think if we eliminate this reaction, if this were truly a negative, would then the forward and the reverse agree? And the answer is yes. So we want to think, well, what could be causing this? And, you know, definitely it could be a type A2 who has an antibody against A1. So what can we do to prove this? We could test this patient's red cells with our anti-A1 lectin. Their red cells, if they are type A2, will not agglutinate with anti-A1 because their antigen is structurally different. Okay? So we're going to test with A1 lectin. And then what we can do with reverse testing is pick 3 different lot numbers of A1 cells and 3 different lot numbers of A2 cells and test the patient's serum, okay, or plasma with all of these cells. Okay, and so if we see agglutination with all 3 A1 cells and no agglutination with the A2 cells, it's basically, you know, and we don't see any agglutination with A1 lectin, it's basically proving that they're type A2. So our next example, cold autoantibody or ALO antibody. Okay, so we look at the forward typing looks like type AB. Let's think. Does type AB have any antibodies to be detected in reverse typing? And the answer is no. We should get zeros or negatives here. And here it looks like they have an anti-B. So they're back typing as type A. Okay, so that's a discrepancy. Once again, these are very strong. We don't doubt them. This one's very weak. So if this reaction was eliminated to a negative, would the forward and the reverse typing now agree? And the answer is yes, because eliminating this, it would look like no anti-A or anti-B, which would agree with our forward typing. So this could be just some other antibody that's reacting with some other antigen on these B cells. And so we could do some tests that we're going to be talking about in more detail later, an antibody screen with a patient control where we test patient with themselves to see, if they're auto-agglutinating or if they're agglutinating with these screening cells. And so basically, this, antibody that they have just happens to be reacting with some other antigen present on these B cells. So when we get these reagent A1 and B cells, they don't only have A1 and B antigens on them, okay? They have other things, and so this happens to be picking up with something else there. Okay. Rouleau, we talked about already. Okay, so once again here, it's typing as AB. Here, anti-A and anti-B, it's typing like O, so that's a complete mismatch. So which do we suspect? These, they're weaker. So if we can eliminate both of these, then the forward and the reverse typing would agree. We want to suspect Rouleau because both of these are looking like weaker reactions, so we could do the saline replacement technique that I talked with you guys about before to eliminate those proteins from the reverse typing. Weaker missing antibodies. Oh man, look at this. So forward typing, no antigens here, look like type O. However, we expect type O to have anti-A and anti-B, and there's nothing here. They're typing like an AB. So what do we want to do? We want to suspect, antigens usually aren't missing. We want to suspect that there could be missing antibodies here. We could check the person's age. But what we could do is, add the serum or plasma with the cells and let those tubes sit at room temperature for 15 minutes. We could even incubate them at a cooler temperature, like in the refrigerator for 5 minutes, believe it or not, and that can enhance reactions. And then centrifuge, and hopefully we're going to get some reactions starting to show up here. So if we get a 1+ even with A1 cells and a 1+ with B cells, then it's telling us that the individual does have antibodies, but maybe they're just not producing enough because they're an elderly person. Okay, so we have a couple more things to do, and what I want to mention to you is in the next unit, we are just going to dedicate the presentation to reviewing these discrepancies. So take them in, think about them, and in our next unit we're going to review them again because sometimes they can be a little tricky for people the first time they hear them. So we're going to move on to a special topic called the Bombay phenotype, which I alluded to earlier. And remember, Bombay phenotype has to do with big H. Okay. So it takes on the name Bombay phenotype because it was first reported in 1952 in Bombay, India, which I think is now Mumbai. And what they found was there were certain members of a family lacked the big H antigen, okay? And so remember, this is usually a dominant, okay? So if they're lacking big H, it means they're little h, little h. This little h gene is an amorph. It does not code for a detectable gene product, so it does not produce the L-fucosyl-transferase enzyme that is necessary to transfer L-fucose to the type 2 oligosaccharide chain to make the big H antigen. Okay, so they type as O. Okay, because they don't have H. Remember, we need big H antigen for A and or B to link onto to make those antigens. Okay, so even if they inherited A and or B alleles, they will not be expressed. Those sugars have no building block to add on to since there's no L-fucose with the H. How do we know something's wrong? They have an antibody against big H, and they have anti-A and anti-B as well. But this antibody against big H would only be in someone who lacks big H. Okay? So this has serious implications for these individuals with regard to transfusion. We can't just give them group O red cells. Okay, because somebody who's a regular group O has the H antigen. And if you think back to when we were looking at H antigen, group O people actually have the most H antigen. So what's going to happen if we tried to give these Bombay phenotype group O? Their anti-H is going to attack the H on these O red blood cells, and they're going to have a transfusion reaction. Okay? So when they are in need of transfusion, it could only be autologous, meaning a unit from themselves that's been stored, or rare donor files. Okay, and so if these families or groups of people are identified, they may actually have them, come into a donor center and donate a unit of blood. And there are special techniques where the red cells can be treated and frozen for up to 10 years. And so they may just store these red cells for them if they need them down the road, okay? Because it's very, very rare, seen mostly in India. Okay, so does everybody get it? They type as group O, but we can't give them O because they have this antibody against H. It would attack any kind of donor O red cells that have H antigen. Okay, so another special topic we're going to get into is called secretor status. And so we talked a little bit about big SE. We were talking about H earlier in this presentation. And so the two alleles here, big SE and little se. And so I will always put a line over the lowercase one so you know it's small s. So remember, this big SE is what controls A, B, or H antigens in saliva and other body secretions. Okay? And so similar to what happens on the red cells, when someone has at least one big SE, H is converted to A or B by glycosyl transferases. It's just going to happen, all that same stuff's going to happen, except it's on a type 1 chain. Okay? 80% of the population are secretors. Okay, so we could be homozygous, meaning we have two big SEs, or we could be heterozygous, meaning we have at least one. Just like big H, this is dominant, so having one is good enough. You're still going to be a secretor, meaning you're going to have those A, B, or H antigens in saliva and other body fluids. 20% of the population are non-secretors, so they're little se, and they will not have antigen expression in their secretions. Okay, so this is an example. If we were meeting in class together, I would say to you guys, let's look at these genes of inheritance and let's figure out, okay, which antigens would be on red cells and which would be on saliva. It's not as much fun doing this virtually because I got to do it myself, unfortunately. Okay, so let's see. They have A, B genes, they have big H, and they have big SE and big SE. Okay, so remember, this is very important for the red cells. Okay, so since this person has at least one big H, they're going to have H, A, and B on their red cells. Okay, now since they also have at least one big SE, what does it mean for saliva? They're also going to have those same three antigens in saliva. It's very hard to write with this pen, sorry guys. All right, now let's look at our next example. Here, A, B, big H. Okay, remember that applies to red cells, so once again, on red cells. But now when we look here, we see little se, no big se. So what does that mean? There are going to be none in saliva. Okay, because that controls what we see in body secretions, and they don't have at least one big S. All right, let's look at example 2. Type O. big H, two big H's, and a big SC and a little SC. So are there going to be any antigens on the red cells? The answer is yes, big H. So remember, O is an amorph. Okay, it does not code for a transferase enzyme to add anything to the H building block, but type O has the most H, so there will definitely be H on red cells. Since there's at least one big SE, there will also be H in saliva. Our second example, OO, HH, little SC, little SC. There's going to be H on the red cells. Will there be H in saliva? No, there will be none. Okay, so I hope that makes sense. Let's go forward to our next slide because I think the next slide has the answers. See if we're right. Okay. Oh, I got to clear everything. Okay. Were we right? Yes. ABH due to this. ABH due to this. ABH due to this. None. Okay. Down here, H for both. Because there's a big SC, there's big H in saliva. Because there is no big SC, there's none. Okay, so hopefully that makes sense. I was right. Okay, so that concludes this presentation. And so remember, in the next unit, it's also going to be on discrepancies, but I'm just going to go over them one more time with you guys because I know they're a little tricky and you can't hear it too many times. Okay, I'll see you next time.